Hello, my name is Claudia Zhang, and today we will be discussing on the topic of determination of gender, more specifically genes versus hormones, and sex determination. But before we go into this, let's start off with a little intro. Humans are sexually dimorphic, which means that males and female organs are distinct from one another. And we have three sets of structures for sex organs, including the gonads. That's the first thing. And it's male gonads, which have testes, and testes produce sperm. And then we have female gonads, and those are ovaries, which produces the egg. Then we have internal structures, such as the fallopian tube in females and the seminal vesicles in males. And we have external structures, or external genitalia, such as the clitoris in females and the penis in males. So how do we determine sex? How does biology determine sex, and how can it all be explained? First, we'll go into genes. The inheritance of Y and X chromosomes determines one's sex. As we can see on the image on the right from Silverthorne, a female would have 2 X chromosomes and a male would have an X and Y chromosome. The female's eggs would only have the X chromosome to give, and the male sperm would either have X chromosome or Y chromosome. And the male is the one that actually determines the gender. If the males donate their X chromosome, then a female offspring would result, and if the male donates their Y chromosome, then a male offspring would result. And that kind of goes into some variations with genes. If there's an XXY gene, because there's still the presence of the Y chromosome, it would constitute a male as a sex. And if an embryo were to only have a Y gene and not an X gene or anything else, then it would not survive, because X genes have essential characteristics that help with development. And then we have a term known as bipotential tissues, and bipotential tissues are tissues before differentiation, when the embryonic tissue cannot be morphologically identified as male or female. And the bipotential gonad will develop into testes or ovaries. The bipotential internal genitalia has 2 pairs of accessory ducts. There's the Wolffian duct, and then there's the Mullerian duct, which proceeds along the female line. The Wolffian duct proceeds along the male line. And we'll get into this now, but starting with the SRY gene. Males have the SRY gene, and the SRY gene is a sex-determining region of the Y chromosome, and in the presence of this, the bipotential gonad develops into testes. And here's another image from Silverthorne, and it kind of shows the 2 pathways that could develop with or without the presence of the SRY gene. If we refer to the right side, for males, they do have the SRY gene. We know that for sure. So first we'll go back to up to 6 weeks, our bipotential gonads are bipotential, but at 6 weeks things start to differentiate, and since males have the SRY gene, they're able to make SRY protein, and this would direct the bipotential gonad to change into testes. The testes would secrete anti-Mullerian hormone, and this would cause the Mullerian ducts that originally existed to disappear, and the Wolffian duct will develop into internal male structures. But for females, it's a little different. Since they don't have the SRY gene, there is no production of SRY protein. And because there is no production of SRY protein, the potential gonad is not told to turn into testes, so it turns into ovaries. And since we don't have any of the SRY stuff, then anti-Mullerian hormone will not develop, and the Mullerian duct would develop. and this would lead to development of female internal structures. Okay. And now we'll be moving on to hormones' roles in developing organs. So these are a couple of hormones involved. We have the anti-Mullerian hormone, the androgen hormone, which comprises of testosterone, and DHT, which is dihydrotestosterone. Okay. So since the SRY gene produces SRY protein, it directs the development of testes, and testes have Sertoli cells, and Sertoli cells secrete anti-Mullerian hormone, as we spoke about before, which causes the Mullerian duct to regress. Genes and hormones kind of have an overlapping role in the development of, or in the determination of sex. And another cell of males is the Leydig cells, which secrete androgen hormones. And androgen hormones include the testosterone and dihydrotestosterone, which is DHT. And they both bind to the same androgen receptor, but they have different responses. For testosterone, testosterone converts Wolffian ducts to make male accessory organs, and it migrates the testes into the scrotum. And then testosterone converts into DHT, which have a role in external genitalia and prostate development. For females, female embryos, like we said before, have no SRY gene, and the cortex of the bipotential gonad develops into ovarian tissue. And in the presence of androgens here, the external genitalia are feminized. So without the anti-Mullerian hormone, the Mullerian ducts develop into female structures, and those can include the upper portion of the vagina, the uterus, the fallopian tubes, and without testosterone, the Wolffian ducts degenerate, and without dihydrotestosterone, the external genitalia take on these female characteristics. So that's why we see the clitoris and the vagina for the female as external organs, and then we see here for males they have the penis and the scrotum, and that's all due to the hormones, the ducts, and the foundation of it all is the SRY gene and the X and Y chromosomes. Thank you for listening to my presentation.